In this lab, we're going to be looking at Mendelian genetics in corn plants, and we're also going to learn how to do a simple statistical test in order to determine whether our expectations under Mendelian genetics match what we see in the lab. So just to give you a little bit of, orient you a little bit to anatomy of corn, um, an individual corn kernel has several different layers to it. The outermost layer, the pericarp, is maternally derived, and so we're not going to really look at this because we're looking at the genetics of the offspring, not the mother. The next layer down in a corn kernel is called the alurone, and this is basically one cell thick, and it can be colored with anthocyanins, which are red or purple pigments. Now, we'll look at the genetics of this, but below that we have the endosperm, which contains carbohydrates that are used by the developing embryo in order to get nutrients and, well, essentially energy from the kernel of corn into the growing embryo before the embryo can photosynthesize. Sometimes if the alurone is clear, so the red or purple pigments aren't produced, we can see the starchy endosperm through that, and that's when we get a yellow. We can see the yellow color. Within the kernel, we also have the embryo, which is going to develop into the next generation of corn plant. Now, the color of the corn kernels is determined by a particular gene that has, for our purposes, has two alleles. The first allele, which we're going to call big R, we'll see that it is dominant. The first allele, big R, is caused by allows the corn kernel to produce anthocyanins in the alurone, and so because of those anthocyanins, we see the color as purple. Now, this gene allows the production and deposition of the anthocyanins. But if there's a mutation in that gene, then something goes wrong, and we don't need to worry for our purposes about what goes wrong, but something goes wrong, and the anthocyanins are either not produced or not deposited into the corn kernel. And as a result, that layer ends up being largely transparent, and so you can see the yellow starchy endosperm underneath it. And so as a result, the little r allele causes the kernels to be yellow. Simply the lack of red color makes it look yellow. Now, we said that this is a dominant trait. So the big R allele causes the production of anthocyanins. Let's think about for a minute what we mean by being dominant. Remember, in past lessons we've learned that dominance basically means, what does the heterozygote look like? Now, in this case, a heterozygote will have a big R and a little r allele, and as a result, it will be able to produce anthocyanins. As a result, it will be purple. And as a result of that, we consider that the anthocyanin allele, the allele that allows the production of anthocyanin, to be dominant, because the heterozygote looks just like the dominant homozygote. Okay? It's only in the little r genotype that no anthocyanins are produced, and because no anthocyanins are produced, we see that yellow color. Okay, so this gene we say it's dominant because the heterozygote looks like one or the other. In this case, it looks like the homozygote that produces the anthocyanins. Now, there's a second gene that we're going to be looking at, and this causes the corn kernels to be wrinkly. Okay, let's actually just take a look at that quickly. Here you can see some kernels are nice and plump, and some kernels are decidedly wrinkly. And you can see that the wrinkly kernels can either be yellow or purple. Okay, so what causes this wrinkly phenotype? Well, the one allele, which we call sugary, and it's the dominant form, converts sugars in the kernel into starch, and it stores the starch as starch in the endosperm. Okay? The other allele fails to do this. It fails to convert the sugars into starch. And so, as a result, when the corn dries out, it loses the water and it dries up, getting wrinkled. Okay? Whereas the dominant form has the mechanism to convert to starch, and that starch causes the kernel to stay plump. Now, this is another case of dominance, and so the dominance allele, the plump allele, allows it to be as a heterozygote, it allows the heterozygote to look like the homozygote dominant. Okay, so we've already looked at this. For this lab, we need to be able to identify the purple kernels and distinguish them from the yellow kernels. And we also need to be able to identify starchy from sugary kernels. Okay, so the starchy ones, remember, are going to be the plump ones that haven't dried out because the starch doesn't dry out so much. And the sugary ones are going to be wrinkly. Now you'll notice here that there are very wrinkly ones, and there are some that look like they have a little dimple in the middle. Don't confuse these. These are different. The ones that have a dimple are still the starchy kind. They're just like the ones without the dimple, phenotypically or genetically. Okay, so in order for them to be wrinkly, to have the sugary kernel, you're going to see them be decidedly wrinkly. Okay, so in this lab, you're going to be counting the kernels that on one ear of corn, and you're going to count the smooth purple, the smooth yellow, the wrinkled purple, and the wrinkled yellow. So you're going to count each of them and fill in a table like this in your lab manual. Now I have done this. Actually, it's just data that I made up for this presentation, but notice that in this case, there is approximately a 3-to-1 ratio for each of these traits. Okay, so here you can see in blue we have the smooth and wrinkled trait, and there's about 3 times as many smooth as there are wrinkled. Okay, in this case, we'd say, well, that looks pretty similar to Mendel's 3-to-1 ratio. Similarly, with the yellow and purple, there's about a 3-to-1 ratio with 3/4 of the kernels being purple and 1/4 being yellow. So, despite having this approximate 3-to-1 ratio, we might want to know, well, is it close enough that we should accept that these are in fact Mendelian ratios, or is something going on that makes it be a little bit different from 3-to-1? And so that's where statistics comes in. Okay, so just to give you a little bit of a background, we're going to imagine that you're flipping a coin, or actually imagine that you're flipping 100 coins. Okay, you throw them all up in the air and then you count how many are heads and how many are tails. Now, because coins are fair, they land 50% on the heads and 50% on tails, we would expect that approximately half of them would be heads and half of them would be tails. Okay, and in fact, this is going to be the single most common outcome. However, notice that it's a very unlikely outcome. It's the most likely, but it's still very unlikely because there's a huge range of outcomes. You might get 50 heads and 50 tails, or you might get 51 heads and 49 tails, or 53 heads and 47 tails. All of these are possibilities. So, but 50 heads and 50 tails is actually going to be the most common. And so the way to read this graph is that the mean value here is the value that is that's for us is going to be the most common. So it's going to be 50 heads and 50 tails. And every time we do this experiment of throwing up 50 coins, we're going to get a different outcome. And each time we get an outcome, we can kind of add to this graph. We can sort of move up on the y-axis of this graph. And so the 50 heads, 50 tails is the most common, most likely outcome. And as we move away from the mean, as we get more and more heads or more and more tails, we start to have less and less likely outcomes. Okay, so for example, 42 heads and 58 tails might be way over here, might be relatively unlikely compared to 50-50. And as we move even further to the right, 35 heads and 65 tails would be much less likely if the coin is fair. If we were to take a really, really extreme case, we might say, well, what about 100 heads and 0 tails? Or 0 heads and 100 tails. Is that possible? Yes, in fact, it is possible, but it's incredibly, incredibly unlikely. You would have to throw up these coins and count them until kingdom come, many, many times until you found a single instance where you had 100 heads and 0 tails. It is possible, but it's very, very unlikely. And as a result, the height on the y-axis of this graph would be extremely, extremely close to 0. Okay. Now, people have done studies of this what we call a normal distribution, and they found that about 70%, just less than 70% of all times you throw it up are within one standard deviation of the mean. We're not going to go into what we mean by standard deviation in this lab, but suffice it to say that it's a measure of how wide this graph is. Okay, about 95% of the data are within 2 standard deviations, and 99.7% of the data are within 3 standard deviations. So as we start to get more and more extreme, we get fewer and fewer possible outcomes. Okay? Now, what statistics does for us is the statistics will help us draw a line and say, you know what? We got say 42 heads and 58 tails. We can ask the question, is that evidence that the coin is biased, or is that just kind of a reasonably common enough outcome of this of throwing up the coins? And doing a statistical test will allow us to kind of draw a line and say, well, this is fairly good evidence that the coin is biased, or it's just normal variation that we would expect, because we don't expect to get 50 heads and 50 tails all the time. We expect to get variation even if it's a fair coin. Okay, so the statistics helps us draw a line and to distinguish between what's pretty likely given random variation and what's so unlikely that actually something might be going on, something strange might be going on. In this case, that perhaps the coin is biased. Okay. Now I do want to just point out that we're going to be using a Chisquare test, and that distribution looks a little bit different from this normal distribution. But the ideas of finding the area of the tails is essentially the same. Okay, so let's look at how this Chisquare test actually works. To do the test, we're going to have to classify our corn kernels as either being, in this case, purple or yellow. And the question we're going to ask is the approximate 3-to-1 ratio that we see, is it very different from the actual 3-to-1 ratio? In which case we might say, well, something weird is going on. Or is it close enough that we would say, yeah, it's not exactly 3-to-1, but it's pretty close. I wouldn't say that anything really weird is going on, because this kind of outcome could happen just by chance. So what we do is we add up all of these four different categories that we've counted, and we classify them simply as being yellow or purple. Okay, and you can see here that we had 1,191 purple kernels and 393 yellow kernels. These were our observed values. So how many did we expect to see? Well, to calculate the number that we expect to see, we would have to take the total that we counted, that 1,584, and multiply them by 3/4. And this is based on a Punnett square. Okay, so we expect 3/4 to be purple and 1/4 to be yellow. So we would multiply 1,584 by 1/4 to get the expected number of yellows. Okay. Now notice that this is you may notice that this is a little bit different from what's called a contingency table, which is often used in statistics if you don't have a formal idea of how many what proportion there should be. Okay. So don't confuse this with a contingency table. We're going to take the expected values based on a Punnett square, based on Mendelian ratios. And we're trying to figure out, are these really different? Okay, so to do the test, what we're going to do is we're going to subtract the expected values from the observed values, square it, and divide by the expected values. And we're going to do that for each of these two categories. Okay, so you can see that I've done it at the bottom here, and because of this formula for the chi-square value, I actually sum them together. And when I do that, I get 0.03. Okay, well, what does that 0.03 mean? Okay, I'm going to show you a chi-square table in just a minute. But you'll see when we look at that we have to compare the 0.03, which is our chi-square value, with a critical value of 3.84. Okay, I'll show you where that comes from in a minute. And we have to ask ourselves, is 0.03 greater than or less than 3.84? Okay, it is not greater than 3.84, so we can see that the difference between the observed and expected values isn't very big. And so we would say the probability of getting an outcome this extreme, and this was not extreme, but the probability of getting an outcome this extreme by chance alone is actually pretty large. Okay, before we move on, let's look at a little bit more closely at this chi-square formula. Okay, you can see that we're subtracting the expected from the observed value. So if the observed and the expected values are very close, if our observed values are really similar to what we expect, then the numerator here is going to be a very small number, perhaps even 0. Okay, we square that number and then divide by e, which is going to be a relatively large number. Okay, as the difference between the observed and the expected increase, okay, that is, as the observations that we make differ more and more from our expected values, then you can see that the chi-square value is going to increase. Okay, so a chi-square value very close to 0 is indicating that there's not much difference between the observed and expected. And therefore we can see intuitively that we should probably accept what we call the null hypothesis, that nothing weird is going on. Okay? As the chi-square value increases, it does so because the difference between the observed and expected values is increasing, and we therefore need to say, okay, something weird is going on. Something besides just random chance is driving this expected value to be, the observed value, to be very different from what we expected. When we did the chi-square test, we compared our observed chi-square value with a value of 3.84. Where does that number come from? Well, if we look at a chi-square table, we find that we can see that there are a number of rows and a number of columns. Let's look at the columns first. We're going to focus our efforts on this column here with p equals 0.05. Okay, this column here shows what the critical values are for chi-squared with a probability of what we call committing a type 1 error of 5% or less. Okay, so what does that mean? Well, biologists have sort of decided that we know that there are, even if a coin is fair, even if nothing interesting is going on, even if we should accept the null hypothesis, we know that occasionally we'll get, just by chance alone, because of the sampling process, we know that we'll get a very, very extreme a very extreme value. Okay, so particularly many purple kernels or yellow kernels, or even a lot of heads and very few tails. Biologists have sort of decided that even though we know, because we know that we can get those extreme values by chance alone, at some point we have to draw a line and we have to say, well, if our value is very extreme like that, then we're going to say that something interesting is going on, something weird is going on. This coin isn't fair, or segregation isn't happening according to what Mendel predicted. And we've decided that even though we say something weird is going on, we acknowledge that some of the time chance alone might give us such an extreme scenario. Okay, and we've accepted that 5% of the time, even though we make that conclusion that something weird is going on, 5% of the time we'll be wrong, just because of chance. Okay, we could get such an extreme case just by chance alone, and we're willing to accept that 5% of the time we'll be wrong. Okay, so we're going to use this 5% chance here, this 5% column, and we also need to understand how many degrees of freedom there are. Okay, we're not going to go into any details about degrees of freedom, except to say that for this test, the number of degrees of freedom is determined by the number of categories of data that we have. Okay, so in our case, we have two categories of data. It's either purple or yellow. Okay, so to calculate the number of degrees of freedom, we take the number of categories minus 1. Okay, so we have 2 minus 1 equals 1 degree of freedom. Okay, so therefore we use the 1 degree of freedom row and the 0.05 p is 0.05 column, and we get a critical value of 3.84, and this is what we compare our chi-square value against. If we find that our chi-square value is larger than the critical value, then we say that the results are significantly different from what we expected under the null hypothesis. Something interesting must be going on. Okay? And we might say something like, there were significantly more yellow kernels than expected. If the observed and the expected values are very similar to one another, the chi-square value will be very small, and we would therefore say that there's no difference between our results and the null hypothesis. So for example, if we had a critical chi-square value of 0.03, which we had in the last example, we might say there's no difference between the data and that expected under the null hypothesis of segregation, Mendelian segregation or Mendelian assortment, whatever the test is we're doing. Okay? Now, I do want to point out that when we test for independent assortment, we're going to have 4 categories, and so you'll see that we'll use 3 rather than 1 degree of freedom. In the second part of this lab, we're going to be looking at independent assortment. Okay, so remember that that's the idea that the two genes are inherited independently from each other. And remember, there's two reasons why they might be inherited independently. The first reason is simply that they're on different chromosomes. And the second reason is that they might be far enough apart on the same chromosome that crossing over is quite likely between the two genes. And so sometimes they appear, a paternal genotype might appear together, and sometimes a paternal genotype might get split up into recombinants. In order to do the chi-square test for this, we're going to look at all four of these categories: the smooth purple, smooth yellow, wrinkled purple, and wrinkled yellow. And we need to figure out our expected values. Okay. Now think about what Mendel's ratios were for his expected values. Okay. Hopefully you can remember that the expected values are 9 to 3 to 3 to 1. So we would expect 9/16 of them to be smooth purple, 3/16 to be smooth yellow, 3/16 to be wrinkled purple, and 1/16 to be wrinkled yellow. Okay, 9, 3, 3, 1. So to calculate the number of expected values, the number of expected kernels, we would take the total number, 1,584 in this case, and we would multiply it by 9/16, okay, to get the expected number of smooth purple. We'd multiply it by 1584 by 3/16 to get the smooth yellow. We'd multiply 1584 by 3/16 to get the wrinkled purple. And we'd multiply 1584 by 1/16 to get the number of wrinkled yellow. After you do this, remember that you're going to have now 4 categories, and so you'll need to calculate your chi-square value for each of those 4 categories, and so you'll get a longer chi-square equation with 4 categories all being summed up, and you'll get your final chi-square value. And instead of comparing it to 3.84, we need to consider that we have 4 categories, and therefore we have 3 degrees of freedom. Okay? We therefore need to compare our chi-square value against 3 degrees of freedom, or 7.815. And if the total sum for the chi-square value that we get is greater than 7.815, then we would say that something weird is going on. The difference between observed and expected values is fairly large, larger than we would expect to happen by chance alone. Therefore, something else might be going on, and we should reject our null hypothesis. If the chi-square value is less than 7.8, then we would say, it's not exactly what we had expected, but it's less than 7.8. And therefore, we can accept that the numbers we see could just be are pretty likely to be generated just by randomness. And so we should accept the null hypothesis that independence assortment is in fact happening. If we're rejecting the null hypothesis would be that independence assortment is happening. Something weird going on tells us that independence assortment is not happening. So maybe they're located close together on one chromosome. Okay, so if you haven't done this lab, then you can make sure that you're able to do a chi-square test like this and understand the logic of a chi-square test like this before we get to the final exam.